Thank you very much, Scott, and a great way to kick off the afternoon of presentations. Hope anyone hasn't got too many revenue forecasts up. Okay. All right, so moving on to our first presentation for the day. We've got Nick Woolfe, who's the CFO of Phylogica. And I'll just read through their brief introduction for them. Okay, so Phylogica is a biotechnology company based in Perth, Australia, and in Oxford, UK, with a world-class drug discovery platform harnessing the rich biodiversity of nature to discover novel peptide therapeutics. Now, I must say, when I first heard it, it didn't really make that much sense to me, but then once I heard it explained to me by Nick, I really understood quite well the potential for this area. So please welcome to the stage, Mr. Nick Woolfe.
Afternoon, ladies and gentlemen. Thanks for that introduction, Steve and Scott. Thanks for that plug. We're delighted to have coverage from Scott. He's got a buy recommendation on us, and he's all three analysts that cover the company have buys on the stock. So as you heard from Steve, we're a biotech company. We came out of a prestigious institute in Western Australia, the Institute of Child Health Research, and also work that was being done in Philadelphia at the Fox Chase Cancer Centre. We're mostly based in Perth, Australia. That's where we have our laboratories and head offices, but we have a small business development presence in the UK, and we're looking now to strengthen that and set up our sort of Northern Hemisphere research base in the UK as well. Steve mentioned that we are based around libraries, biological libraries. These derive from some of the most extreme bacteria in the world, and I'll cover that in just a second. And we've integrated these libraries as a source of novel drugs and also as a source of understanding disease pathways into a one-stop shop for big pharma. And our business model is around commercializing that through fee-for-service deals, where we have reach-through rights, though. In addition to revenue up front, we get reach-through rights through milestones and royalties to anything that comes out of our libraries. And we've got some household names in terms of partners we're currently working with, listed at the bottom there, and we've signed all of these deals in the last two and a bit years. So we've only been in the commercial phase since 2009. Now, we're in a sweet spot for the industry, this sort of outsourcing of drug discovery. Pharma companies over the last few years have been cutting back on their in-house research and their in-house drug discovery, because of the poor productivity, the slowness, the general ethos within pharma has become less conducive for drug discovery, and so all of these companies are now looking for companies such as Phylogica to be able to provide their drug discovery engine. So what is our Phyloma platform? Well, peptides. Peptides are nothing more than fragments of proteins. Proteins have been a very successful biological class. Antibody drugs, for example, fall into that. But peptides have a unique capability, being a small fragment of a protein, to do things that a big molecule can't. And it's not a new market. The market is already $13 billion. Insulin, for example, is a peptide drug. It's growing, and as a market, both in terms of development and in terms of in the development of new drugs and in terms of sales, it's growing twice as fast as the traditional small molecule pharmaceutical market. So these pretty postcards here show some of the extreme environments from which we source bacteria. Now, we're in the genetic age now, so we don't just chop up bacteria, we chop up their genomes. So we have in our libraries almost 40 genomes sourced from some of these extreme environments, and we synthesize one long genome like a mega-organism, and then we exhaustively fragment that up genetically, and that is the basis of our libraries. So we have 10 billion peptides from fragmented genomes in our libraries. It's a very rich source of drug candidates, because nature has evolved to make these proteins and peptides drug-like, to make them interact with biology in whatever they do. And we source our bacteria from the organisms marked in orange here. Now, humans are right over here, and we often get the question, well, why don't you just chop up the human genome and find drugs from there? And the answer is because there's a lot of junk in the human genome and a lot of structures that are similar. If you really want diversity, you need to look in the bacterial kingdom and the ancient archaea. these are organisms that diverged 3 billion years ago along this tree here. So there is more diversity in the genomes of these organisms than there is between humans and fungi, for example. So I'm not going to give you any data today, don't have time, but just one snapshot very quickly. This shows us looking going into our libraries to try and find antibiotics against a bacteria called Acinetobacter. And these are what we call primary hits. So they haven't been optimized or polished or anything like that. They're just primary hits. We just screened against bacteria and pulled the turquoise colors are our peptides. And the lower the bar, the better the inhibition of bacterial growth. Now, these are two marketed programs. This is two marketed antibiotics. This is amoxicillin, for example, which I'm sure most of us have had treatment with. Now, we've got primary hits that are more effective, have better antibiotic activity than these marketed products. So our business model is to take our libraries and integrate them, as I mentioned, into this one-stop shop. So all these chevrons show the areas of expertise that we have. So we have our libraries. We can do what's called high-throughput screening. So if a pharmaceutical company comes with a target, we can it takes us 6 to 9 months to interrogate our libraries and pull out optimized potential drug candidates. We can then improve those drug candidates through maturation. We go in terms of we can look at where evolution has sort of varied the sequence and helped to improve the binding activity and the potency. And we can do things to make it very drug-like, like improving the half-life, so it's not eradicated from the body so quickly, and stabilizing these peptides. If you've got a peptide and a target, you want to make it so you can stabilize it with various linkers, and we've got access to all that to make us really a one-stop shop for peptide drug discovery. But unlike a lot of biotechs, we're focusing on what we're good at, and we're focusing in what we're good at is the drug discovery end. At this point in time, we're not pretending to be a clinical company. We don't want to be exposed, as some of the references that Scott made, to companies where clinical costs go up. There's also the binary aspect of drugs obviously failing in clinical trials. So we generate revenue straight at the get-go from doing deals. People come to us, we say, Big Pharma, what are you having trouble drugging? There's a verb to drug in our industry. What are you having trouble drugging? Let us see if we can, let us, Phylogica, interrogate our libraries and see if we can find something interesting. And they pay an upfront fee, a technology access fee, then they pay our research costs with a markup, then there are success fees if we pull out peptides that meet the objectives of asked of us, and then it goes into their development, so our pharma partners do the development, and we reap the benefit through milestones and royalties. So to date, we've signed these 4 deals, and the 2 key messages from this slide, before I run through each in turn, is A, the economics of our deals. The financial terms are getting richer and richer. So there's a single product deal at the top. We've just done our first multi-product deal. Potentially up to 15 candidates could come to market from this program. And the second key point is that we have delivered on everything asked of us to date. These are in chronological order of when they were signed. Roche came to us and wanted a peptide that could target the brain, could cross the blood-brain barrier, and we achieved our objectives there. They're now evaluating our peptides in San Francisco with a view to exercising what is an option to license. And then we get, obviously, then we're in the milestone schedule of payments. The MedImmune collaboration is around finding antibiotics against one of those hospital-acquired superbugs called Pseudomonas. Massive market, real need with all the drug resistance that's being developed, and they've come to us for that. The Pfizer collaboration, it's a therapeutic vaccine against an inflammatory target, and we've totally nailed that one, and we delivered exactly what they want now, and they've got our peptides in animal studies with a view to exercising their license this year. A very big milestone for us. And the Johnson & Johnson collaboration, we signed it literally the week after Christmas and on the 30th of December. Big deal. It's our largest upfront payment from this deal. It's potentially got many options to expand. They could be, and come back. They could be funding us for up to 5 years under the current collaboration. So we've got 4 lines filled in. And we're looking to fill in the rest of these. And as Scott said, our guidance is that we will have another couple of deals in this financial year, and we're on track to do that. In fact, the next one is pretty close to getting over the line. And we're also now talking to more mid-tier companies as well. So from a financial aspect, our revenue from 0 a couple of years ago went to 2.5 million in FY11 and is on track to double again in FY12. Our cost base is very static. It's around $7 million. We use a lot of robots to do the work, so we've invested some capital expenditure, but we don't need to increase our headcount at this point. We've got cash in the bank today of a little over $4 million, in fact. That was the 31st December figure. It's enough to take our company through to sustainability in the next 12, 18 months. So news flow wise, and obviously it's difficult to give guidance on deal doing, but we take that plunge and we're feeling pretty comfortable with the guidance we've given. We've got new pharma partnerships happening. We've got the potential deal expansion with Janssen, and some significant milestones on our other collaborations. So from a summary, just very briefly, we've got a pretty strong management team. Some of you may know Doug Wilson. He's our executive chairman. He ran medicine for Boehringer Ingelheim, a big German pharmaceutical company. Our CEO, who can't be with us today, is also our scientific founder, Dr. Paul Watt. He spends most of his time overseas doing talking to our pharma partners. Myself, my background, I'm from the UK. If you haven't guessed from my accent, was with a company called Oxford Biomedica and actually worked on Scott's side of the fence before that as an analyst. So in summary, we have this world-class platform for peptide drug discovery. We've validated that platform both in our in-house work and through our partnerships. We are genuinely on a very steep curve from the commercialization aspect, and we are looking to break through into cash flow sustainability in the not too distant future. We are now talking to some of our existing partners as well as new partners about some much larger collaborations, which would really tie us closely with these companies, potentially with a view to a trade sale, which we've always cited as a potential exit for our shareholders. That's the end of my presentation, but I will just mention that in terms of our ticker, I haven't mentioned that, it's PYC. Our market cap today is $25 million. We've got a single class of share. We have a pretty strong institutional holding at the top of our register. About 25% of our shares are held institutionally. We've got a couple of big US biotech funds, and a fund in Sydney called Naos Asset Management, who owns, Naos actually owns 13%. They're an emerging companies fund. And then we've got a number of high net worths that have been with us since the IPO in 2005. I've got some material at the back, and you'll see the stand. Please come and approach me later. Thank you so much.